out what's happening there. So just hold on one second. Okay, hello everyone. Welcome to Concordia University's Fourth Space. Thank you for joining us for the next installment, kind of a special one really, of our Closer Look series. This is where we look behind closed doors and spaces into projects, things you might not know about or get a chance to see at Concordia. Today, we're going to be taking a closer look at the Sacher Lab, and I'll let our Sacher Lab, a remote team, explain a little bit more about that. But just to help situate you, we are streaming to YouTube live from Concordia University, and we are located here on unceded Indigenous lands in Chichage, Montreal. Okay, and just a little note of caution. We are hosting this as a live stream event, so things can happen. Video connectivity, audio issues, things like that. That's actually kind of part of the fun. So just bear with us. We're also running this as a Zoom meeting, and at the end, after this, left of the live stream part, because we're very lucky to welcome the YMCA Steampunk crew here this afternoon, we're going to do a little Q&A with our microphones so that you can ask our friends at the Sacher Lab some questions. So think about some questions after we do the tour. Okay, that's enough. That's way too much from me. It's now my pleasure to pass us over to the remote team. We've got Mahsa Mehranfar, who's a PhD candidate in biochemistry and cellular and biochemical basis of rare diseases at Concordia. And Brett Cox joining us, who's also the coordinator of sustainability and research at Concordia University. I'm going to pass it over. Brett, Mahsa, it's great to see you.
Thank you so much, Doug. I appreciate it.
Hi, Doug.
Thank you for having me. I can start with a bit of an introduction. First of all, Doug, you're looking good. Are those new glasses?
It happens, yeah.
Yeah. Okay. Well, it's
glasses actually, yeah.
It's been a while. Anyway, I can start with a bit.
You were here in Fourth Space. This time we've sent Brett to the lab.
Yeah. This is my first time remote, so it's cool to see you on the other end here and see how this works. All the magic behind the scenes. So I'll give a bit of an introduction as to why this came to be. So I'm coordinator of sustainability and research. And so I was mandated to help support a variety of different projects that are leading and advocating for the various actions in Concordia's sustainability and research action plan. If you just Google Concordia sustainability, you can learn about the action plan, the different streams. I support the research stream, and part of that stream, one of the projects that was brainstormed was around developing tools and various supports for helping researchers embed sustainability into their research practices. And with that, my supervisor and I brainstormed a project specifically looking at research practices in wet labs. And so with some funds, we collaborated, we built relationships with 3 different labs in the Faculty of Arts and Science, 2 in chemistry and biochemistry, and then 1 in biology. And then we had 3 different grad students, Mahsa being one of them, test out sustainable research practices in our lab. And there was also a bit of a budget to buy equipment to help further that. So it's been a great learning experience for me because my background is in social sciences. So this has been all new for me. And I've really appreciated Mahsa and her work. And it's been really fascinating.
So this has been the third episode, I guess, of the different labs that were involved in the sustainability practices project, right? This is the third, right?
Exactly, yes. Yeah. So with that, I can pass out to Mahsa, introduce herself, and give us a tour.
Thank you so much, Bert. Hi, Doug.
Hello.
So to just give you a brief introduction about what we do in the lab. So basically our lab is a biology and molecular biology lab, and I'm a biochemistry student. So basically in the lab I work with proteins, those big molecules that have a specific function in cells, and our body is made of a lot of different type of cells. So for my project specifically, I'm looking to understand a function of uncharacterized protein called Tango2. So it has a very nice name. It's not Tango, it's Tango. And for my work, I'm trying to actually isolate the protein from the cellular environment and have it in the lab and use it for some biochemical assays. This is one purpose of my study. And another thing that I'm looking for is that I want to understand where this protein is localized within the cellular environment. So as you know, a cell consists of different organelles, and these organelles are within cytoplasm, a little bit of cell biology here. And I'm looking for its localization. It's very important to know where this protein is. So if I wanna like share some of my research with you, so the first thing I did in the lab is to isolate this protein from bacteria. So what I do is just I express this protein in bacteria, and I use affinity chromatography, which is a specific method to purify the tagged protein that I have in the cell. And once I have that protein in the lab, I can use it for a lot of purposes. I can do a lot of interaction study, binding study, enzyme assays, and so forth.
So can I ask, what was the name of that process? Sorry, you just mentioned.
So like basically the process is protein purification, purifying protein from like a mixture of millions of proteins. So what I do is just I tag this protein. It's like having a microchip on a human. So I tag a protein with engineering. I put a histidine tag at the very end of this protein. And then that tag has affinity to a specific column that I use in the purification system. So like metal and magnet. So my protein is his tag, and the column that I use is a nickel NTA, has a nickel NTA bit. So these have affinity, they attach to each other. So that's how I purify this protein from cell. And I have one single protein that I'm gonna show you in the lab, so I use it for my experiments. It's kind of like very versatile method when it comes to biochemistry.
And the lab that we're in this, we know of it as the Sacher Lab. And what building are you in? I forgot to get a sense of where we are.
So we are in Loyola campus SP building. I don't know what's the abbreviation of that.
I don't remember.
So yeah, it's SP building, and we are on the 4th floor.
The 4th floor. And the lab itself is quite small. Or maybe it's not.
Yeah, this is the main portion of the lab, but we have some facilities that we use. Most of our equipments are there. So for example, like we have a microscopy center that like we do microscopy as dot center. We have tissue culture facility that is actually upstairs, and we grow cells, we work with the cells there. And also we have a mass center, it's mass spectrometry center at the 1st floor. So it's just like the main work is being done here, but we use different facilities to access the equipments.
Okay. Thank you.
So basically, if I want to show you, like, if I just want to give you like a brief introduction about what I do. So I have, for example, this bacteria grown, this bacteria that are expressing my protein. And I actually suspend the pellet of the bacteria in a specific buffer. And then like what I do is I use this active purifier for protein purification. So basically in real-time experiment, I load the whole cell lysate into this column that we have here. So this is the column that I talked about. So my protein is a tag protein. It is histidine tag. And this column is nickel in NTA. So histidine and nickel, they have affinity towards each other. So once I actually put this cellular extract on this column, I have these mobile phases that flows liquid over this. So all of this protein, my protein of interest, it stays in the column. And then I can just visualize the process with this. So as you can see, this is the chromatogram that actually detects my protein at a specific UV wavelengths, because proteins have this UV absorbance. So at UV to 80, I can see this is the protein peak. So it will be collected in small fractions, and I use that fractions to detect the protein, and I run STS page to see where the protein locates. So basically, like here are two fractions that I collected from ACTA, and this is my protein. So I visualize that with Kumasi blue staining. So this is a dye that actually reacts with the protein, and then we can see, okay, like if the protein is one single band or not. So this is the purified product. And I like once I had that, I use it for biochemical assays, biophysical assays that I do, and a lot of versatile experiment can be done with this.
Okay. Amazing.
Yeah, it's very nice to see proteins. And then another thing that I do is just to separate mitochondria from cellular environment. So mitochondria, as you know, is a powerhouse. So it provides energy for the cells. So like one of these assumptions that I have for my project is that this protein is involved in mitochondria, probably is in mitochondria and do something with metabolizing fatty acids. We don't exactly know, but we are looking for that. So, but we must confirm that this protein is in mitochondria. So what I'm doing is that I grow human cell lines because I'm working on the human version of this protein. And I grow the cells and I pelleted the cells. These are approximately 30 million human cells.
30 million.
30 million. Can we get a close-up of what 30 million human cells
this
looks
is the
like?
volume of 30 million human cells, very tiny cells. And then, like, I have to break down these cells to extract mitochondria. So mitochondria is a kind of, it can be like actually segregated by differential centrifugation. So like, I want to show you how the process works a little bit. You might be interested to see that. So what I'm gonna do, I'm gonna add a specific buffer. This is homogenization buffer to these cells. And I'm going to resuspend the cells by using this micropipette in the buffer.
And sorry, what was this process called again?
Homogenization.
Homogenization.
Yeah, the process of breaking down the cells. So like these cells are having organelles surrounded by a plasma membrane. So I'm trying to break down the plasma membrane so that I can extract mitochondria.
Okay. And so just so maybe I clarify a little bit, or maybe you can tell me if I'm wrong. So this protein that you're studying, the tango protein,
Right.
it's a bit of a mystery protein. And so what you're doing right now is you're trying to figure out where it is located.
Exactly.
Okay. It's a little Where's Waldo for this protein.
It is.
And you suspect, your thesis is that it's located in the mitochondria.
So yeah, like we think that it's a cytoplasmic protein basically, but I mean, we suspect that a portion of this protein is in the mitochondria. So by using this cool apparatus, I'm trying to homogenize the cells. So it's just like I'm giving that very gentle strokes with this. So it breaks down the plasma membrane that actually surround the cells, but not mitochondria. Just avoid bubbles.
And how often would you do a process like this? Is this your daily process that you do?
Yeah, it's just like I do that pretty much once per week because I'm trying to optimize this experiment to get like a good like publicable figure for my paper. So yeah, like I'm trying to optimize, and actually it's been a long time I'm doing that. So it's just I have to optimize concentration of detergent, buffers, like the concentration of initial cells that I put for this that I use for these experiments, and then the concentration of antibodies. So there are a lot of variables that I need to optimize to get the best result that I look for. Once I have done that, so I would transfer the cells to a small tube. So this process should be on the ice. So that's why I can't.
Okay, so these are all kept together on ice
Yeah.
to maintain their temperature.
Yeah, to just stop the cellular metabolism. We don't want to, and then we have proteases that degrades protein. So we want to stop the protease activity as well. And then I'm looking for some microtubes. I have them here. So I'm gonna pipette. So I have to transfer that like that.
And so we've homogenized everything. That's what you were doing in the little tube, and now this hopefully is a separated.
So these are this is a mix of broken cells and unbroken cells. So like I'm gonna separate unbroken cells because those are the things that I don't want. I just want broken cells. And that's why I do a low speed centrifugation.
Okay.
So I need
So this was the prep before the centrifugation.
That's true. So this is a balance that I need for the machine.
And then the centrifugation will separate the broken cells and the unbroken.
Exactly. Thank you so much. I'm really happy that you understand. And then I centrifuge for 5 minutes at very low speed. So this is 1000 RPM, and I press start. So this takes 5 minutes, and then we can see a phase separation.
Okay.
Okay. So once this is, yeah.
Go ahead.
Yeah. So I was just thinking maybe I can show how.
I was curious though, Mahsa, in terms of the sustainable practices that you were looking at with Brett in this process. What kind of sustainable processes had you identified in the lab?
So basically, it's just like at the beginning, I realized that we are consuming a lot of plastic tubes. So it's just I do like my best efforts to just reduce this plastic waste and replace the plastic tubes with glass ones wherever it's possible. So it's not, for example, like now it's not possible to not use plastic microtube because I'm doing centrifugation, and glassware, I mean, it's not compatible with this centrifuge. But sometimes like that I have I work with some buffer that are not hazardous. I can replace, for example, these tubes with these glass tubes, for example, and these are recyclable, and also it can be washed and reused. So I just bought like different sizes of these tubes. So for example, this tube has a cap. It can be actually, the material can be stored in the fridge as well. So and also we have these ventilation vials that can be replaced.
Were there any problems switching from using plastic tubes to using glass?
So basically, like using these glasswares, like it reduces this plastic waste, but it cannot be used in when we are doing some sterile work. For example, if we are growing yeast or bacteria,
Yep.
we cannot use these glasswares. But like for other stuff that don't need like a sterility, so we can easily switch to glassware, and like we put them in the aluminum basket, and they go to the dishwasher, and like that can be reused. The dishwasher that we have, so both washes the tubes with detergent and distilled water afterwards. So that is ready for like my lab members to be used, right?
Yeah.
So this is what the first thing I come up with, and also like we have these small ones that are very cute to work with. So for example, for Bradford assay, that I use 1 ml of Bradford dye. So I use these tubes. These are very cute. I really like working with them, and these are sustainable.
I can imagine with all these different iterations, all these variables,
Yeah.
all of the testing that you're doing, and all of the sterilization that's required to keep everything, on track, that
Right.
that sustainability, this seems like actually maybe it might cause a lot of obstacles, because what do you like? Yes, of course we can switch to glass in some ways, but there's sterilization. But then there's also the washing of those glass things. So
That's
there's
true.
still water
That's true.
waste into, so it's kind of a bigger question of like, what are the best modes? What are the most sustainable? It's like a whole new variable in all of your science experiments.
That's true. That's like a drawback of that. We reduce the plastic waste significantly, but at the other side, like there is more water consumption and detergent consumption as well.
Yeah. Do you guys have a sustainable solution for water? Were there any sustainable measures that you took for water reduction?
We are using bead baths instead of water baths.
Oh, yeah.
So like it's at the other side of the lab.
Shall we go see?
Yeah, sure.
Okay. Ooh, and we get to see the other side of the lab. I know that one of the other labs that we were visiting, they really zeroed in on the water use, that they really noticed water use being something that they could address. But here, what are you showing us here?
So basically, like we have these traditional water baths that actually work with water, and basically because we have water and I think 42 degrees temperature, we need to add these biocides that prevent microorganism growth to this water. So this is not environmentally friendly. It stays in the environment, and also like we need to frequently change the water for the water bath, so it's consume water, right? And then for the cleaning of the water bath, because sometimes we need to maintain this, we need to clean up everything. So for the cleaning, we need to wash the system and rinse it with water and stuff. So that consumes more water. So what we are using now, so it's just we replace this water bath with this bead bath. So these are the bead baths that we have. We have more of these in our tissue culture facility, where we should actually warm up media bottles and PBS every day, for example. Like there are a lot of users there. So this is, the company claims that these are more energy efficient. So like the energy that is consuming to heat up these beads is, as the company claims, is 1/4 of the energy needs to actually heat up that water bath. And also these are recyclable. So once their lifetime is done, they can be easily recycled. And also the maintenance of this piece is very easy. We need to just, every 3 weeks, we need to just spray ethanol 70% on these bits, and that's it. Don't need to wash them.
Oh, so no water at all, just a 70% alcohol.
And we don't need to add biocides that are not environmentally friendly. And like this, like one challenge that I can see is that these beads are a little bit expensive, but this is just one-time purchase.
That's it, as opposed to all the water. So maybe I'll just back up a little. For those who aren't here in the lab, the water baths are another way that they're keeping everything at temperature. And so not only is the water bath not only do you need the water continuously added to keep it at that temperature, but you also need to keep that water completely sterilized, which is why you're adding that extra solution, which is also environmentally harmful. So now with these beads, you're limiting water and you're limiting the use of that environmentally unfriendly substance.
Yeah, and it's consume less energy as the company.
And less energy. Look at that, triple threat.
Yeah. And also these are the aluminum baskets that we use for our dishwasher. So we put the tubes are here, and then we wash them in the dishwasher when once they are full. So I think like the frequency of washing these tubes is every 1.5 months or every 2 months. So once it's full, we really wash them. So we try to consume less water in that respect as well.
Did you have a question, Doug?
No, actually, I was wondering if what point maybe, Bertie, if you can, when we could switch to more of a Q&A, because maybe there might be some questions here from our steampunks.
Yes, amazing. I think we could probably pivot to that. If Mahsa, if you have anything else you wanted to talk about in your process to end us off before we open it up to Q&A.
So there is one more step with the cells, and I'm done with that.
All right, so we have one more step with the cells. So maybe we'll finish that journey and then we'll open it up in case we answer any questions with this last step.
I know we had something in the centrifuge.
Yeah.
Yes, we're going back to the centrifuge.
Okay, we see two phases here. So these are actually unbroken cells, and this is the phase that contains mitochondria. So I'm going to separate the upper phase into another tube, and then I centrifuge it with high speed. So that's the way that you can pellet mitochondria, and that's ready for the experiment. So I'm going to carefully just separate the upper part.
So what's in this file now will be...
So this is the crude mix, which contains mitochondria. So I'm gonna grab this. So I'm gonna increase the speed to 10,000. So 10 times higher speed now.
So it's going back in the centrifuge?
So it's gonna like pull it at the bottom of the tube.
Okay, so you're pulling it down.
Yeah. As we increase the centrifugation speed, so like every particle that is inside in the supernatant will be settled at the bottom of the tube.
This thing's going really fast. I'm gonna try to hold the mic so you can hear it, guys. That motor's going.
Yeah. So this takes 5 minutes, and then we can go for any question that you have.
Wonderful. So that's the end of the process that Mahsa goes through. So if we have any questions in the space, we can certainly open it up.
Okay, so we're gonna close up our live stream to YouTube now, and then I'll just say farewell to anyone joining us on YouTube, because this will remain available on our YouTube channel. You can visit it later. And in the meantime, for those of you on Zoom, I know there's a couple of people on Zoom, and then our steampunkers here in the space, we can have some questions with Mahsa and Brett.